Okay, so hopefully that wasn't too painful. I realize it might have been. If you even put your name on the piece of paper, you will get some points, that I promise you. In addition, next time I say that there's something I'd like you to go look up, I hope you take it seriously. But don't panic. Hopefully that's not too painful. Let me just tell you very quickly the answers, okay? So last time we saw that the half-life of DNA was on the order of 220 million years. And I challenged you to go out and find out how to measure it. And at least one person came to my office hour with a solution, which is to heat the DNA. So you can heat it up and to a high temperature, measure the half-life at the higher temperature, and extrapolate back to the lower temperature. And you don't have to provide me with a lot of equations to show that that's true. But if you just write heat on problem number 2, I will accept it. Okay? But again, if you have your name on a sheet of paper with your UC ID number, you'll get at least some points. Okay? Okay. All right. We're back to normal stuff now. I want to pick up where we left off last time. We were talking about structure of DNA and reactivity of DNA. And we saw last time, oh geez, what happened here? One moment, let me just find this. Okay, we saw last time a lot about Watson-Crick base pairing and structures of DNA in its bDNA form. Now, one thing I got asked about immediately after the lecture was, ethidium bromide is commonly considered to be a carcinogen. This molecule over here that we discussed as an intercalator of DNA is also a carcinogen. It's a molecule that causes cancer. And that's the reason why when we work with it in the laboratory, over here, we're exceptionally careful to keep it off of our skin. Okay? Now, it intercalates into DNA. And DNA intercalators can help to initiate carcinogenesis, can help to initiate cancer in the following ways. Number one, it distorts the structure of DNA. I showed you that on the previous slide over here, where I showed you how the structure of the DNA has to unwind to accept the intercalator. So it distorts the structure of DNA. Number two, it places a hydrophobic functionality in the center of the DNA. This hydrophobic functionality can inappropriately attract transcription factors to the DNA, setting off incorrect transcription. Okay, so those are two possible modes that this can start to cause, that this can start to initiate improper cell responses that will eventually lead to cancer. And I want to talk about cancer today. It's one of the prime topics in this class. It's something we're going to spend quite a bit of time discussing in many different contexts. And we'll certainly be talking about it in the context of DNA. Okay, so any other questions about what we saw last time? That was a really good question. Okay, last thought about ethidium bromide. Although it is definitely a cancer-causing agent, and it is something you definitely do not want to ingest, and you definitely want to keep it off your hands, I don't want to exaggerate its carcinogenic potential. It's also used as an antibiotic in sheep. I don't know if it still is, but it was for a while. It does have some other, so it's actually fed to sheep. I don't know why sheep. But in any case, as a tumor-forming agent, its activity is rather modest, especially compared to the other cancer-causing small molecules that we're going to see very shortly. Okay? All right, well, let's dive right in then. So last time I was showing you that DNA likes to form Watson-Crick base pairing. This is C's binding to G's, A's binding to T's. And we saw that GC's Watson-Crick base pairs have 3 hydrogen bonds, and ATs only have 2. Okay, now, if we know that, that AT base pairs are weaker than GCs, there's a very simple rule that we can use to estimate the strength of any 2 DNA strands sticking together. This rule is called the Wallace rule, and I'd like you to memorize it. Okay, the Wallace rule tells us that the approximate melting temperature for a DNA sequence is equal to 2 times the number of AT base pairs plus 4 times the number of GC base pairs in degree C. This melting temperature is where 50% of the DNA is no longer forming a double-stranded structure. Let me explain. So when you do a UV-Vis scan of DNA, this is here's what you find. Okay, so this is absorbance on the y-axis and wavelength in nanometers on the x-axis, and this is single-stranded DNA, and then this is double-stranded DNA in blue. And to get this, you simply add higher temperature. So at higher temperature, the DNA strands melt apart. In other words, they separate out into the 2 single strands. And so at 82 degrees, this is the single-stranded, and at 25 degrees, this is the double-stranded DNA. Notice that the absorbance at 260 nanometers is higher for the single-stranded DNA than the double-stranded DNA. So you can use that change in absorbance to follow whether or not your DNA is single-stranded versus double-stranded. And so you can do this while at the same time you ramp up the temperature. And at about 50%, and so here it is, pure double-stranded DNA, lower absorbance, and then here it is at a higher temperature where it's entirely single-stranded DNA. And the approximate 50% part, where 50% is melted, is called the melting temperature. And again, you can estimate what this melting temperature is using this Wallace rule formula. People in chemical biology laboratories use this Wallace rule formula on a daily basis. Okay, certainly in my laboratory, and certainly in probably 5 or 6 other laboratories here at UC Irvine. So I'd like you to memorize this rule. It's incredibly useful. Now, you're probably wondering, big deal. So I know whether or not something melts, whether or not it forms double-stranded DNA. If you know the temperatures that it forms double-stranded DNA, you can start to design structures made out of DNA. Let me show you. Okay, so here are structures of DNA where it's single strands of DNA that are now hybridizing against each other and forming elaborate patterns, such as this pattern here. And here's an atomic force microscope image of this double-stranded DNA, and you can see it's forming this exact cross-link pattern that was designed using something just a little bit more complicated than the Wallace rule, which I asked you to memorize. Okay, it gets even better. Check this out. Okay, so this is work done by Paul Rothamund and colleagues and coworkers at Caltech, and he's using the Wallace rule to design DNA that folds up into happy faces over here. Or check out this map of the world written out of DNA that's been folded up with itself. Okay, so that Wallace rule that I asked you to memorize is actually pretty powerful. You can develop whole structures of this stuff. Now, exactly what these structures of DNA are going to be useful for is not 100% known at this point. There is sort of a frontier in chemical biology, building structures out of DNA and then trying to do something useful with them. I've only seen one paper in 20 years of staring at these beautiful pictures that has convinced me that maybe there might be something useful about this. And in that paper, a DNA structure like this one, not the happy face, but something elaborate, was used as a delivery vehicle that bound to the surface of cells and then dislodged a drug therapeutic. And it's possible that our future might feature many more of these sort of examples of nanometer scale structures that are designed by the people in this room to have specific properties, such as binding to specific cell types, unloading cargoes at specific times, etc. This is a really exciting frontier, and I encourage you to think about it in your proposal preparation because it's an area that's kind of wide open for creativity. Okay, so in addition to those sort of macro structures of DNA that we saw, short stretches of DNA can also fold, and there's a couple of canonical structures that we're going to see time and again. One of these, for example, are called hairpins. One of these is called a hairpin, and so this consists of a sequence that folds back on itself. Notice that it satisfies all of the Watson-Crick base pairing requirements, G's to C's, A's to T's, and it forms something that looks kind of like an old-fashioned hairpin. Okay, and it looks structurally like this. This is the X-ray crystal structure of what it looks like. And again, we're going to see this quite a bit. Okay, so DNA has a propensity to fold on itself. It wants to form Watson-Crick base pairs. It wants to form Watson-Crick base pairs with other sequences. It wants to form base pairs with itself. And so for this reason, DNA is rarely found as sort of an unwound, it's rarely found as single-stranded DNA, for one thing. And furthermore, it's rarely found in this sort of canonical bDNA conformation that I've been showing you, where it's this nice right-handed coiled double helix. Rather, in cells, we typically find DNA in a wound-up configuration called a supercoil. So a supercoil is where you take a coil, such as this old-fashioned telephone cord, which I'm sure is unfamiliar to everyone in this room. But back in my day, kids, we used to have these. And it would form these sort of supercoils, and it drove you nuts. You know, you'd constantly be on the phone trying to untangle the darn thing. In fact, actually, it was kind of a nice thing to do, because if you're on the phone with a tense conversation or something, it gave you a task to take your mind off of the annoyances that you were dealing with on the phone. But anyway, so this is called supercoiling. And this is an example of a DNA plasmid, which is a sequence of DNA that forms a circle. Okay, so this is a nice plasmid DNA. And again, we rarely find it in this sort of bDNA where it's completely unwound configuration. Rather, it likes to twist up. So here's a little twistiness, here's more twistiness, even more twistiness, and then finally, even the most twistiness. Okay, so that's really the structures of DNA that we find. For short sequences, we find it wound up with itself to form hairpins. I showed you that structure first. For larger sequences, like plasmids, we find it supercoiling and twisting itself up. And then for even larger sequences, like eukaryotic genomes, which I'll show you on the next slide, it gets even more twisty than that. Okay, so, before I get to that, here's why it has to get so twisty. This is one of the benefits of having it coiling up on itself. This is just the DNA in a single E. coli cell. This is a classic picture of that DNA where really tour de force microscopy was used to lyse the cell and spew out all of its DNA. And you could see it's just an enormous amount of DNA for such a small cell. And human cells face a similar compaction problem, right? The human genome, it would be roughly 1.7 meters if completely unwound. So this property to want to wind on itself, to coil up with itself, is actually a really important one. And so when we look at human DNA, we find that it's compacted up into chromosomes. I'm sure, hopefully, this is not an unfamiliar concept. In fact, actually, I showed you examples of chromosomes from, I believe it was guinea pigs when we talked about bromouracil earlier this week. In any case, so here's some images of human chromosomes over here, and here's how it's compacted up. So the DNA, here's the bDNA that we've been looking at over here. I admit this is a terrible rendering because it looks like it's a single helix. Note that it's lacking the major groove. Lacking the minor groove. This is one of my pet peeves about artistic depictions of DNA, but I'm going to let it slide by at this moment. In any case, so here's the regular bDNA. The bDNA is going to wrap around a protein structure called a histone, and this is going to act like the spools for thread, and then these histones are going to coil up, and those coils are going to coil up further until eventually you get to something that's massively compacted into a chromosome. Now the problem, of course, and this is the problem if you're on the phone as well. So imagine you're on the phone with one of those old-fashioned telephone cords. What you find is that it's very hard to untangle the DNA without disconnecting the cord and making a little break in it. Okay, and that's one of the annoying things about supercoiling of old-fashioned telephone cords. Similarly, with DNA, where you have this 1.7 meter long object and a 20 micron long cell, there has to be a solution to uncoil the DNA. And the solution is to make transient breaks. So short-lived breaks in the DNA using an enzyme called DNA gyrase. And here's an example of this. This is a DNA gyrase that acts kind of like scissors. Okay, so notice that it's a dimer. The two arms down here can open up, and the thing can just grab on to the DNA. And then introduce two breaks in the DNA, allow the DNA to relax, to uncoil, and then it gets rejoined. This turns out to be a Achilles heel for bacterial cells, for cells in general. In other words, it's a spot that can be targeted with antibiotics. And we'll be talking about these. We'll be talking quite a bit about different ways that antibiotics work. So antibiotics are pharmaceuticals, therapeutics that are given to patients to eliminate bacterial infections or fungal infections. In this case, this is a really effective antibiotic that's given quite a bit. This is the antibiotic Cipro, which I am sure many of you in this classroom have taken at one point. I read somewhere that like 85% of American women come down with a UTI, a urinary tract infection at some point. The first line of antibiotic used against that is often Cipro. And Cipro works by inhibiting the DNA gyrase of bacteria. Okay? Here's another one as well, another inhibitor as well. Okay, so let's talk a little bit more about these bacterial plasmids, because I want to transition into a discussion of biotechnology and cutting and pasting DNA and large scale. Oftentimes DNA is transferred amongst organisms using plasmids. Plasmids are short circular stretches of DNA. They need to have, all plasmids need to have two properties. Okay, they must have sequences that encode two items. Item number one is an origin of replication. The origin of replication, abbreviated ORI, is the spot that somehow convinces the cell that's taken up the plasmid to start replicating that DNA. Okay, so that origin of replication kicks off replication of the plasmid. Without that, the plasmid would just be there and it wouldn't get copied and it wouldn't get passed on to the next little guy. Okay, so that's absolutely essential. The other essential thing is that the plasmid has to confer some advantage onto the new host. Okay? In other words, the new bacteria has to take it up and say, oh yeah, this is useful. Otherwise, the plasmid will get quickly shunted aside because cells are under a lot of pressure. They have a lot of work to do, and they have a limited amount of resources, carbon, nitrogen, oxygen, things like that, that are available to do all of the priorities that they have. Okay, and that's kind of a long-winded way to say that this has to confer some resistance, oftentimes, to some sort of antibiotic. Okay, so resistance markers are sequences of DNA that encode a protein that confers resistance. Okay, so for example, you can have a resistance marker that encodes resistance to the antibiotic tetracycline, and this gene will work by actively pumping tetracycline out of the cell. Okay, so when the cell takes up this plasmid, it's going to synthesize this pump that goes to the surface of the cell, and then every time it gets tetracycline, it just pumps it out of the cell furiously, and that allows the cell to live. So only the cells that have the plasmid will survive an onslaught of the antibiotic tetracycline, which again is a very common antibiotic that I imagine a few of you have encountered. It's often used, for example, I believe for acne treatment. Okay, here's some other classic examples of other antibiotics that are used in my laboratory and other chemical biology laboratories as selection markers for drug resistance. The way this works is we'll coat the cells, the bacterial cells, on a plate, and the plate has agar, which I'll show you the structure of very shortly. It's isolated from seaweed. It's basically just a polymer, and inside this agar plate we'll have some concentration of one of these antibiotics. And so the only colonies, and each one of these circles is a colony, that appears on this plate are bacteria cells that have taken up the plasmid, because now those cells are resistant to the antibiotic. Okay, so here's another way that this can work. So antibiotic that's commonly used is chloramphenicol. Chloramphenicol inhibits the ribosome. We talked about the ribosome before. The drug resistance gene encodes an enzyme called chloramphenicol acetyltransferase, or CAT. And this enzyme transfers an acetyl group to a primary hydroxyl of chloramphenicol. Okay, so here's the acetyl group in acetyl-CoA, and it's going to get transferred to this primary hydroxyl in a reaction. that essentially disarms the chloramphenicol, preventing it from binding to the ribosome and allowing the cells on this plate of chloramphenicol to live. Third, very common resistant marker. Okay, so I've shown you tetracycline, we've talked about chloramphenicol. Third one. The third one are beta-lactam antibiotics of this sort. Notice that this is a beta-lactam. A lactam, of course, is a cyclic ring that has an amide bond in it. And this is beta because it has 2 carbons, alpha, beta. And so that's the that's the origin of the beta-lactam nomenclature, which I know we talked about in 51C. Hopefully you encountered as well. In any case, beta-lactamase is a enzyme encoded by the beta-lactamase gene that, that confers the ability to hydrolyze this amide bond that's part of the beta-lactam ring. And this is a very common gene that's found out in the environment. So you can probably scoop up some dirt over here just outside Rowland Hall, and you can readily find this beta-lactamase gene. And so for this reason, medicinal chemists are constantly making new antibiotics that avoid that environmental drug resistance that's sort of omnipresent. Okay, so for example, here are 2 different kinds of beta-lactam-based antibiotics. And notice the structural differences. This one has this benzyl functionality over here. This one has a carboxylic acid and a phenyl group instead. And so all those little differences affect the ability of the drug resistance enzyme, the enzyme conferring drug resistance, to bind to the antibiotic and hydrolyze this amide bond. Maybe this carboxylate sticks into the protein and prevents the binding, and that's a useful thing. Okay, so we're constantly on the hunt for new antibiotics because the antibiotics we have seem to allow very rapid evolution of drug resistance. And so there's a constant need, really, for our society to develop new classes of antibiotics that are more effective than the previous generation. And in the last 10 years or so, there's been a real renaissance of research in this area to develop even more effective antibiotics. Okay, let's get back to our discussion of DNA structure. I showed you structure of plasmids. Here's structure of eukaryotic genes, eukaryotic DNA that's wrapped around nucleosome, etc. I don't have very much more to say about that. Let's take a closer look, however, at the structure of these histones. So the histones are these hexameric proteins shown here in yellow and green, where in green, these are positively charged residues. Okay, so those are residues whose positive charge can interact with the negatively charged phosphodiester backbone of the DNA. Charge-charge interaction, nice long-range interaction. This wrapping up, though, basically hides the DNA and prevents it from being transcribed. When it's wrapped up around a histone, it can't be read out and used for transcription. And so basically, whether or not the histone is wrapping up things, it controls transcription and controls packaging. So these proteins over here are very tightly regulated as to whether or not they're going to be binding to the DNA. And one easy way to do this regulation is to acetylate the lysine side chains. Okay, and I'll show you that on the next slide. Okay, so first, this is the structure of lysine. Lysine has a primary amine, and here's lysine with an acetyl group. What do you think the charge is of something as a primary amine at neutral pH, which is the pH roughly of the cell, approximately? So it has a primary amine functionality and neutral pH. What is its charge? I'm a very patient guy. Positive. Very good. Okay, good. So if this is bare lysine, it will have a positive charge. And if it has an acetyl group over here, it is back to neutral. Okay? So this guy, positive charge, acetyl group, neutral. So that controls whether or not that lysine amino acid of the protein interacts with the DNA. If it's positive charge, it's like a homing beacon for DNA, right? The DNA is negatively charged. Two of these want to stick together. If it's acetylated, however, it's going to be neutral, and the two are not going to want to interact with each other. Here's one that's even wilder. In this case, you're taking the primary amine of lysine side chain and turning it into a secondary amine, or tertiary amine, or even a quaternary amine. And when you do this, you're making the lysine side chain fixed as a positive charged. Okay, now I should say it's not fixed permanently. It used to be thought that it is, but now we know that actually this is a reversible modification, as is this acetylation. Okay, so this case, it's binding to DNA, and then when you get rid of these methyl groups, it's back to, it's still binding to DNA, but then it can get acetylated, so it's no longer binding to DNA. Okay, so there's a whole series of different modifications to the surface side chains, the surfaces of the histones. And all of these modifications have important consequences. Okay, so for example, some of these modifications, like these larger ones down here, direct the histones into the proteasome, which is basically the garbage disposal for the cell. And so those get flushed away and thrown into the trash. And then others, like this phosphorylation of a hydroxyl functionality found on the surface of the DNA, can regulate the structure of the histone as well, and perhaps interfere with its binding to the negatively charged DNA. Okay, so all of this stuff is tightly choreographed. There are enzymes that add each one of these modifications highlighted in blue on this slide. And those enzymes are going to control its binding affinity for DNA, and in turn control whether or not the DNA is hidden or available for transcription. And you can imagine this is very tightly choreographed by the cell. If anything gets in there to mess stuff up, all kinds of havoc can be wreaked, right? Because the cell has to control, turning on, specific genes at specific times. Right? You would not want, for example, a muscle cell to suddenly start growing, I don't know, neurons or so, the genes that are required for neurite growth or something like that. That would be really bad. Okay, so everything is very tightly choreographed at this level. All right. There are, of course, small molecules that inhibit these histone deacetylases. I shouldn't say of course. This is actually a discovery that was made by Jack Totten, who was a graduate student when I was a graduate student in the same lab where I was. This discovery was made when I was a graduate student in the laboratory where I was getting my Ph.D. And in short, this is what the N-acetyl-lysine surface looks like of the histone. So here it is sticking out. Here's the N-acetyl. And then here's Trapoxin, which looks remarkably like this lysine, this acetyl-lysine, right? These look very, very similar. Maybe a slightly different number of carbons, but it looks very, very similar. And so this is going to be one possible way to design inhibitors of enzymes, which is to mimic the substrates. Okay, so this is the starting material for the histone deacetylase, the enzyme that chops off this acetyl group. And then this compound over here is going to inhibit that deacetylation. There's more written about this in the book. But in any case, these two look very similar. And so that substrate mimicry, that mimicry of the starting material, is a very common way to inhibit enzymes. It works really well. We're going to see that time and again throughout this class. Okay. I want to change gears now. I've shown you all the cool things that you can build out of DNA. I want to talk to you next about how to actually synthesize the DNA so you can build these things. Okay, if you want to make happy faces, or maybe you want to make the first frowny faces out of DNA, hasn't been done before to my knowledge, you're going to have to know how to synthesize the DNA so that you can make that happen. Okay, so I'll talk very briefly about DNA synthesis in the lab, but first I want to talk to you about DNA synthesis by the enzyme DNA polymerase. Okay, so DNA polymerase takes a single-stranded piece of DNA called the template and adds a second strand of DNA to that template. Okay, now all DNA polymerases found on the planet have a common mechanism, and they all require a starting primer strand that gives it sort of a running start. Without this running start, the enzyme doesn't know where to begin. And this is actually a very useful property, right? You don't want DNA polymerase to come along and start synthesizing random, bits of pieces of DNA here and there. And it turns out that this is one that's been exploited quite a bit. And I'll show you some examples of that in a moment. Okay, so this starter is called a primer. It forms, again, double-stranded DNA with the targeted template. And then DNA polymerase lengthens this priming strand in a 5' to 3' direction. In other words, it grabs onto this 3', it adds the new 5', etc. Okay, so this direction here is also common to all forms of DNA polymerase found on the planet, 5' to 3'. The starting materials here are nucleotide triphosphates. Structures, so it's the nucleotides that I showed you earlier, but with triphosphates attached to them. Okay, and basically what it's doing is, again, it's taking the green primer strand and lengthening it, as shown by this arrow over here. So this is a classic experiment that was done that applied this principle to crack the genetic code. The genetic code is the code by which sequences of DNA spell out the amino acids. Okay, and this was back in the 50s and early 60s. There was this enormous mystery about what that code actually was. Okay, it was like this, unsolved major problem. And Marshall Nirenberg at I think it's Rockefeller, but I might be wrong about that. All right, Marshall Nirenberg used this property to crack the genetic code. What he did was he synthesized templates that were long strings of particular DNA sequences. Okay, so he made a long string of A's, for example, and then he looked at not what was synthesized by DNA polymerase, but downstream what was made by ribosomes when you give them a long string of A's. And then by doing that, he could figure out what the genetic code was. Okay, so again, DNA polymerase requires a template to lengthen an existing strand. Only RNA polymerase can start from scratch. Okay, so RNA polymerase is kind of an exception to this rule. DNA polymerase requires a priming strand. Reverse transcriptase, which takes RNA and synthesizes DNA, we saw that earlier, also requires a primer and deoxynucleotide triphosphates. RNA polymerase is kind of a special case. By the way, any questions? You guys feel free to interrupt if there's anything that comes up, okay? Anything that's unclear, you want to know more information about it, don't hesitate to stop me, okay? Yeah?
Does the primer get replicated as well? Or is
the primer gets extended, but it doesn't get replicated in the mode that I'm showing you. When we talk about PCR, we'll show that it actually can get replicated. So, okay. Yeah, that was a good question. Other questions? Yeah?
If you wanted to hybridize like a piece of a primer with
yeah.
a mutation in it,
yeah.
how many like base pairs do you usually need to?
Okay, these are great questions. Okay, awesome. I'm delighted you're asking. I forgot to ask for names. What was your name?
Paul.
Paul and?
Anthony.
Anthony. Okay, so Anthony's question is, how many base pairs of DNA should you have to get to use as the primer to get DNA polymerase going? Okay, and it kind of depends. Okay, so you want DNA polymerase to pick up a specific gene or a specific sequence of DNA within a complex mixture. And so if you want to pick up a specific gene in the human genome, you need a primer that's at least 18 base pairs in length. Okay, that's kind of a magic number. Okay, so 18 base pairs means that you're uniquely encoding one and only one gene in the human genome. Okay? Thanks for asking. It's a good question. On the other hand, if you want to do this at a lower temperature, you can use the Wallace rule and get away with maybe a shorter sequence. Okay, maybe you don't need such specificity. Maybe your mixture, your starting population is less complex. Okay, thanks for asking, Anthony and Paul. Okay, let's move on. Okay, so here's a close-up view of what I've been telling you about in hand-waving examples. We're now zooming down to the level of atoms and bonds. That, of course, is what really thrills me. So here is the primer and DNA polymerase. Not shown is that, of course, this primer is forming a double-stranded DNA to the template strand, and also not shown is that the template strand must have an adenine over here to hybridize to this thymine. Okay, in any case, the starting material used here is a deoxynucleotide triphosphate. Note that it's deoxy at the 2' hydroxyl. Okay, and here is the 2', here is the triphosphate functionality. The leaving group in this reaction is going to be diphosphate, which is used very commonly in biology as a leaving group. This is nature's tosylate or mesylate that you learned about back in Chem 51C. This thing works really well as a leaving group, and it's one of the reasons why we're going to see it quite a bit as a leaving group. As all DNA polymerases, all enzymes that use diphosphate as a leaving group absolutely require a dication to bind to this diphosphate, and the requirement is for magnesium in DNA polymerase. Okay, so actually a very common problem that I see in my own laboratory when a newbie shows up in the lab and they have trouble with their DNA polymerase, 9 times out of 10, it's due to low concentrations of magnesium. Okay, and there's a lot of ways to get low concentrations of magnesium. So a little tip. Okay, so here's magnesium. What is it doing? Magnesium is a Lewis acid that's chelating to this diphosphate and stabilizing its negative charge. Doing this makes it a better leaving group, right? This means that if it goes out into the solution, it doesn't require some massive rearrangement of water. It's already been stabilized. It's at a lower potential energy than it would otherwise be. Okay, so here's the role of DNA polymerase, and I'll show you structurally what it looks like in a moment. DNA polymerase brings together the 3' hydroxyl of the primer together with this incoming nucleotide triphosphate, and then sets up a nucleophilic attack on the phosphorus of the nucleotide triphosphate. Note too that there is a second magnesium ion in the active site. This second magnesium ion does 2 things. Number 1, it helps to stabilize the alkoxide formed when this 3' hydroxyl is deprotonated. Okay, so notice that it's forming an ion pair relationship with this alkoxide. Number 2, it actually increases the nucleophilicity of the lone pair that's going to do this nucleophilic attack over here. Okay, so what magnesium is doing here is it's making available, better available, this lone pair for an attack by helping promote the deprotonation of that hydroxide. If the hydroxide is deprotonated, there's more lone pair that's available for the attack, right? Make sense? Okay. This forms an intermediate. There's a collapse of the intermediate. Analogous to what we saw on Tuesday when we looked at the hydrolysis of DNA, exact same mechanism. That intermediate is not depicted. I'm showing arrows over here. But again, we've looked at that intermediate before, so I feel comfortable leaving it off of this slide. Okay, any questions about this mechanism? Yeah?
Where does the magnesium come from?
Ah, great question. What is your name?
Nick.
Nick. Okay, Nick's question is, where does the magnesium come from? Magnesium comes from the food you eat. It comes from, you add it to the test tube. So typically we'll add magnesium chloride or magnesium sulfate directly to the Eppendorf tube, tiny little test tube that we use for these reactions. Okay, but in humans, we're eating all kinds of food that has magnesium in it. Okay, so but it's absolutely essential. Okay, so without the magnesium, this reaction does not go. Okay, makes sense because I'm showing you what a key role it plays. Okay, let's look structurally at what this actually looks like. This is an enzyme that again has a number of different orthologs or homologs. These are enzymes that do more or less the same thing. Reverse transcriptase synthesizes DNA from an RNA template. The enzyme TAC is a DNA polymerase that's used quite a bit in research for a tool called PCR, which I'll talk about in a moment. But all of these enzymes have a right-handed structure. Okay, and here's what this structure looks like. Okay, so here's a right hand over here. Here's my right hand. And it's grabbing on to the DNA. Okay, so the DNA is in red and orange over here. And here's the enzyme grabbing onto this DNA. Now what happens is during the synthesis, the DNA threads itself through the crook formed by my thumb and palm. Okay, and as at a certain when the correct nucleotide triphosphate binds to the priming strand, the newly synthesized strand, the enzyme can then close. When it closes, the palm and the thumb get closer to each other. Palm, thumb, and fingers. Okay, so it closes a little bit like this. Each time that closes, that brings the magnesiums up to the triphosphate, setting up formation of the covalent bond that I showed on the previous slide. Okay, so each time the hand closes, that's one nucleotide that's been added. Okay, so let's do this. All right, we have this, one bond. Okay, now let's do a couple more. Bond, bond, bond. Okay, now here's the deal. This enzyme is really cranking. It's actually going to do 1,000, up to 1,000 of these per second for some of these enzymes. Okay, so that's like, too fast to see, really. Okay, so this enzyme can really turn over very quickly. And actually, this is actually something that my laboratory has directly observed. We actually have watched one of these enzymes cranking over, and we've watched differences as we add different substrates to the enzyme. It's a really absolutely fascinating series of experiments. Okay, so all these enzymes use a common mechanism. Again, the enzyme doesn't close until it gets the correct nucleotide triphosphate that binds to it, and at that point it closes. So in fact, actually, the rate determining step for this enzyme is actually the arrival of the correct nucleotide triphosphate. A's to T's, or DATP to T's, DCTP to G's, etc. Okay, I want to tell you a little bit more about why I'm in such love with this enzyme. This is a dream machine. I like fast cars and I like fast enzymes. This one is really amazing. So check this out. Imagine that double-stranded DNA was about 1 meter or so in diameter, okay, running along the length of this room. Okay, so we got some DNA running through the room. All right. If that was true, DNA polymerase would be about the size of a FedEx delivery truck. Okay, including the polymerase. There's some other replication machinery that I'm leaving off for now that's involved as well, but it'd be roughly the size of a FedEx delivery truck pulling up right here. Okay, but here's the thing. This delivery truck would be racing along at about 375 miles per hour. And that's how fast DNA polymerase is going in scale to the DNA. Furthermore, it's making about 1,000 covalent bonds per second, which is insanely, insanely fast. And in addition, I haven't talked about this yet, but there are other subunits of DNA polymerase that are providing an error checking and correction function, such that the enzyme is making to scale 1 error every 106 miles or so. Okay, which is extraordinary. Okay, 375 miles an hour and 1 error every 106 miles. Okay, this is truly remarkable stuff. You could read more in this reference down here. Okay, now, because this enzyme is so efficient and so superbly specific at getting the right Watson-Crick base pairing, this has been used very commonly in lots and lots of laboratories. Chem bio labs, molecular biology labs, biochemistry labs, forensics laboratories, all kinds of labs use DNA polymerase. And they often use it to amplify up copies of DNA using a technique called the polymerase chain reaction invented by Kary Mullis amongst others. The way this works is you start with some target sequence of DNA shown here in purple. Again, we'll call the target the template. Okay, so that's the template DNA that you're going to amplify. Now, there are going to be 3 steps to this PCR reaction. In step 1, we heat the DNA up to high temperature, say 95 degrees. And as we've discussed earlier today, DNA, when it's heated up to high temperature, goes from double-stranded to single-stranded. It falls apart. In step 2, the solution is cooled down, and that allows the primers, shown here in green and blue, to anneal, in other words, hybridize to the single strand of DNA. Note that these two purple strands don't find each other. The concentration of template is very, very low. In fact, you can get down to just a few copies of DNA. So they never find each other. They're like, lost from each other after the heating step. But you have a high concentration of these green and blue primers that can grab on to the correct sequence of DNA. That motion targets DNA polymerase, drags DNA polymerase to synthesize a specific stretch of DNA. And then that's done in the third step, when the primers are extended using, again, DNA polymerase, DNTPs, magnesium chloride, and a temperature of 72 degrees. To make this work, we use a special DNA polymerase that likes to run at 72 degrees. It's a type of polymerase called TAC, which is found in hot vents, hot springs. It's in an organism that's found in these hot springs that has evolved to operate at this temperature. And so at 72 degrees, the enzyme starts cranking. At the lower temperature, it's not working. At the higher temperature, it stops working. But at 72 degrees, it's love and life. Again, this is Celsius. This is pretty warm. It starts synthesizing this black strand of DNA. If you do this process a whole bunch of times, each cycle you get a doubling of the amount of DNA. And say you do this 30 times. You get a huge amplification for some target template of DNA, some target sequence of DNA. Okay, makes sense? Any questions about this? I'm hoping I'm not telling you anything you don't already know. PCR is now taught at like high school level and stuff like that now. So, okay. Summary. Right-hand rule. We looked at this business about the magnesium 2+ stabilizing the nucleophile. We've looked at this already. Why don't we move on? Okay, so DNA polymerase is also a terrific target for inhibitors, and reverse transcriptase inhibitors have been very, very important compounds for stopping synthesis of DNA. And there's many reasons why you'd want to stop synthesis of DNA. To treat, for example, cancer, where cells are dividing uncontrollably, if you could shut down the replication of DNA, you'd have an effective way of stopping cancer. And in fact, actually, childhood leukemias were stopped in their tracks back in the late 70s through the wonderful research of one of my scientific heroes, the great Gertrude Elion, shown here. Gertrude Elion was born in 1918. Her parents wanted her to become a nurse. Okay, so they sent her to college and they said, go and become a nurse. She actually wanted to become a chemist. When World War II broke out, she was given her opportunity. Okay, so during World War II, the men were sent to the front to fight, and there were a lot of opportunities that were available to women that weren't available before then. And she's one of those people who took that opportunity. She joined Burroughs Wellcome, where she worked with George Hitchings for her entire career. She's one of these people who spent her entire career at a single company. And together with George Hitchings, she discovered this class of compounds that inhibits DNA polymerase, and ended up actually having a major impact on childhood leukemia. She's a true superstar of science. Okay, one last thought. Gertrude Elion never received her Ph.D. She went on to receive a Nobel Prize in the 80s for this work. She did it through sheer force of will, through her determination to contribute something. And I highly, highly recommend an interview of her, and I'll even put it up on the board over here. There's, if you want to learn more about her, there's this terrific interview of her that's in a documentary that I recommend. Okay, so the documentary is called Isaac. Can you see this okay, Natalie? Okay. Isaac Newton and Me, and the director is the great Michael Apted. I could actually have a whole class just on Michael Apted's documentaries. But in any case, she's interviewed in this documentary, and she talks about the incredible pride and just joy that she felt when she would visit children's hospitals and she would see kids being treated for the first time with her compounds, and how transformative that was in the life of these kids. These are kids that were slated to die at very young ages, at the ages of 10 and 12, and that were suddenly getting cured by these compounds. Okay, let's take a closer look and understand how these compounds work. Okay, so she invented a series of inhibitors of DNA polymerase that look like this. Okay, so this is one called AZT. It's also used as a anti-HIV compound because it inhibits reverse transcriptase. It looks kind of like a DNA base. It has a deoxy at the 2' hydroxyl, but in place of the 3' hydroxyl, there's an azide. Okay, so what happens is this gets taken up and phosphorylated to give a triphosphate, and then DNA polymerase attempts to use it as a substrate. But what ends up happening is, instead of a 3' hydroxyl, there's an azide here. And the azide caps the synthesis of the new strand of DNA, preventing it from being lengthened. Okay, similarly, this is DDC, another compound that's used in the treatment of HIV and also leukemia. And instead of a 3' hydroxyl, it has a hydrogen there. And so again, it gets used by DNA polymerase, and the polymerase can no longer lengthen the nascent strand of DNA. Okay, so both of these shut down DNA synthesis. I'm simplifying things a little bit. There's another class of compounds, non-nucleoside analogs. These are nucleoside analogs that are also used against HIV. But that kind of gives you a taste for what Gertrude Elliott did. She had a major, major impact in the fight against viruses and in the fight against leukemia. And I actually had the pleasure of meeting her once in my life. The day I was getting my Ph.D. at my graduation, she was receiving an honorary Ph.D. from Harvard, and I just shook her hand. That's about it. I didn't have any profound conversation with her, which is to my regret. But truly remarkable woman, true superstar of science. I can't say enough about her. I can have a whole lecture about her. Why don't we move on? Okay, so that's DNA synthesis in the cell. Oh, question over here. Yeah.
So these nucleotides that she synthesized, how did they deliver them to the cancer cell?
Oh, this is such a good question, and I'm so glad you asked. What is your name? Bhavin? Okay, so Bhavin's question is, how do you get the compound to the cancer cell? What we're going to see time and again is that cancer cells are actively eating up every little bit of nutrient that they can find. They will just be devouring stuff that's around them. And so for this reason, they and other dividing cells will preferentially take up drugs like these that are fed to the patient, that are injected into the patient. Okay? So the problem, of course, is that there are other cells in the body that are also dividing and that will unfortunately take up these chemotherapeutics and also end up dying because their DNA synthesis will be impaired. So, okay, great question. Thanks for asking. Okay, so I don't have very much to say about chemical DNA synthesis. I think it's an absolutely amazing topic. This is one of those areas of organic chemistry that is a true triumph. Okay, so we're going from strength to strength today. In DNA synthesis in the laboratory has been so optimized that we're at the point where we get 99.9% yields for reactions. Okay, this is really the ultimate goal in the quest to do organic synthesis. And it was set in line by the great Gobind Khurana, who won a Nobel Prize, but also Bob Lessinger and Marty Carruthers. These guys deserve a Nobel Prize because this really did kick off a revolution in biotechnology that was made possible by the synthesis of DNA to make primers, which in turn allows PCR, which in turn allows smiley faces out of DNA and all those other great discoveries that we've been talking about. Okay, now I just want you to skim this topic in the book. Don't get too worked up about it. In the end, we have these machines that look like this, where you have a bunch of bottles down here that inject that you can use. You can program a computer up here to inject reagents directly into flow cells that have the DNA sequence that you're trying to synthesize. So you do this using solid support-based synthesis, where the nascent strand of DNA is attached to some sort of bead, and you basically flow in the reagents one after another and couple the correct nucleotide directly onto the DNA sequence. It's a little more complicated than that, but here's what you need to know. If you want to synthesize any sequence of DNA that's 150 base pairs, 150 bases or less, you get on the web and you call up some lab, probably in Texas or somewhere like that. And there will be a whole warehouse of machines like this, and you enter into some form on their website exactly the sequence of DNA that you want. And that sequence will get ported to a machine that looks like this. And there'll be this warehouse just filled with these machines. And then there's a bunch of technicians on rollerblades that are going to be running around and keeping the machines fed with reagents. You won't see any of this, because at the end, you'll get a FedEx package with your DNA sequence probably in a couple of days. Some of these, I think, are even overnight, right? So overnight, you're going to get your sequence of DNA perfectly cleaned up, purified, delivered to you with 95% or 99% purity, depending on how much you decide to pay for it. You know, it'll be perfect every time. You won't even have to think about the chemistry. I love that. That's the goal of organic synthesis. The goal of organic synthesis is to make this totally turnkey so that we can then use this thing to answer biological problems, which is what I want to talk to you about next. Okay, here's one example of using this amazing ability to do DNA synthesis to address biological problems. You can print sequences of DNA on microscope slides. Okay, so here's a machine that's nothing more fancy than, I think in this case, it's an inkjet printing device. Okay, and it's going to be printing out little oligonucleotides one after another on these identical microscope slides. So each square down here is a microscope slide. Okay, and across the surface of these microscope slides, we're going to have a bunch of different sequences of DNA that have been printed down onto specific spots. Okay, so what this is going to do is this is going to give us an array of different sequences of DNA, each one capable of hybridizing, forming Watson-Crick base pairing to a different other sequence of DNA or RNA. Okay, this is a technique called the DNA Microarray. Okay, so here's the way this works. Let me just show you what it looks like. Okay, so now I'm zooming in on the microscope slide. Okay, so here's what it looks like. Each spot over here is a different sequence of DNA, and you set up the hybridization such that you end up with a fluorescently labeled sequence. Okay, so each spot has a different sequence of DNA. If there is a complementary sequence in your sample, then you will see fluorescence. Okay? So in green, that tells you that your sample has this particular sequence. Okay? And it's known exactly what the sequence is in green that's down there. Okay? Because you've synthesized the complement and put it down right there. Okay, so if, I don't know, let's just say this is the gene that confers resistance to beta-lactam antibiotics. Yes, you have that gene because you're seeing green spots right here. In practice, this can be made a lot more complicated. Okay, let's imagine now that we have two samples. One that we label with red sequences and one that we label with green sequences. Okay, this allows us to compare all of the different DNA sequences present or RNA sequences present, comparing them against red versus green. Okay, and let's take a closer look. Okay, so each oligonucleotide hybridizes to a different mRNA transcript where the one sample is labeled in green and the second sample is labeled in red. Okay, here's the way this works. On sample 1, you use reverse transcriptase to convert all these RNA into DNA, and you add the green fluorophore. In sample 2, you use reverse transcriptase and label all those with a red fluorophore. Okay, so sample 1 is green, sample 2 is red, and then you add both of those to the DNA microarray. The ones that are in green are telling you, oh, that gene is upregulated in those types of cells. The ones that are in red is telling you it's upregulated in the other types of cells. And where red and green overlap, you see yellow. Okay? So this allows you to compare experiment versus control, and the possibilities are endless. For example, you can look at cancer versus non-cancer cells, virus infected versus normal, G1 phase of the cell cycle versus S phase. I'll explain these phases in a moment. Young cells versus old, drug treated versus no drug, etc. And so in the end, you get these massive arrays where you get a huge amount of data. Each one of these red spots, that could tell you, oh, that's a gene that's upregulated in, say, cancer cells or virus-infected cells. And then the yellow ones, those are ones you don't have to worry about. The green ones, though, right, because that's the same in both types of cells. The green ones, however, are ones that are upregulated in the, let's say, the non-virus cells, okay, and downregulated in the case of the cancer cells. So you can see in one very simple experiment, relatively simple, little complicated, you get out an enormous amount of information about the transcription activity of an entire cell. Again, this is all set in place by DNA synthesis, chemical DNA synthesis. Okay, so here's an example of this, a specific example. The example I'm going to use is the small molecule FK506, also called tacrolimus. This is a immunosuppressant that was discovered by the pharmaceutical company Fujisawa, hence the name FK. As an immunosuppressant, it is given to patients who had an organ transplanted. Okay, so this was kicked off sort of a revolution in the area of liver transplantation, for example, when it first became available in the 80s, late 80s and early 90s. Okay, so if you give this drug to your patients who have just had the liver transplant, they will not reject the new transplanted liver, okay, because their immune system is suppressed. You can kind of see the problem with that approach, though, right? I mean, the immune system is suppressed. That means that if they get a cold, they're going to be in big trouble. Setting that aside, okay, there's other things that you can do. Let's try to look at what pathways are changed when we feed cells this compound. In doing so, we can start to identify the pathways associated with the immune response. And so here's a classic experiment done by the company called Rosetta Informatics, now Merck Pharmaceuticals. And in this experiment, they have two kinds of cells. One kind of cells are green. They were treated with the green fluorophore, so the transcripts were treated with the green fluorophore. And in the red, so these have no drug added to them, and in red, those transcripts were treated with the compound I showed on the previous slide called FK506. In yellow, there's a whole bunch of different compounds. Check this out. In green, over here, this is a protein, this is a gene that encodes a protein that must be turned off when the compound is added. Okay, notice that it forms a bright green spot. Over here, here's one that gets turned on. It forms a red spot. The yellow ones, the orange ones, let's not worry about those so much. Okay, but you can imagine doing all kinds of analysis of this stuff. The bioinformatics side of these things, the computation to analyze this stuff, fascinating, okay, and a really exciting area of computer science research. Okay, does this make sense? Okay, so one experiment, you get the whole pathway that's being targeted by this drug. Okay, I'm not telling you very much about the pathway now. Maybe we'll talk about it later. Yeah, question in the back. Okay, yeah, so in practice, you have a laser that scans across the surface of this microarray, and then you have a CCD camera that captures the intensity of each spot. Okay, I'm hoping this is blowing your mind, because it certainly blows mine. Okay, so now I wanted to change gears a little bit and talk to you about how to analyze DNA, sequences of DNA. Before I do, I'm looking at the time here. I'm going to run all the way up to the 10 before the hour, but I just want to tell you I have some good news for you, a little weekend treat. Thanks for bearing with me with the quiz. The midterm will only cover through chapter 3. There'll be no chapter 4 in the midterm. Okay? So when we come back on Tuesday, we'll be talking more about, don't get ready to go. I'm not going to stop now, but I just want to let you know. We'll be talking more about DNA, and then we'll go into RNA. But the midterm on next Thursday, a week from today, will only cover through Chapter 3. Okay? All right, let's get back to our discussion. I want to switch gears very slightly. Earlier, I alluded to agarose, which is used in those Petri dish plates that I showed you earlier. Agarose has this structure down here. It's isolated from seaweed, and it is a polymer that is tightly cross-linked to form a net. Okay? You can actually form little bricks formed of this stuff, and then you can apply the DNA to one end of the agarose gel. So here's your little brick called an agarose gel, and you add your DNA to one end of this gel, and then you use electrophoresis to push the DNA through the polymer. Okay, recall that DNA is negatively charged, so it will be attracted to the positively charged terminal, positively charged electrode, the cathode, at one end of your electrophoresis apparatus. Okay, so again, you have these wires coming out. They're going back to some source of electricity, some power supply, and that's pushing the DNA through the gel. The DNA will get separated out then on the basis of its size. Okay, so bigger DNA is going to get caught up in this complicated network over here, whereas the little pieces of DNA are going to find it easier to flow through these little pores. Okay, so in practice, what this looks like is this. I think I've already shown you one of these agarose gels. This is the top of the gel where the big pieces are. Here's the bottom of the gel where the little pieces are. Again, the big pieces have not migrated as far because they got stuck in the interstices of the DNA. Frequently, we add what's called a DNA ladder to one lane of the gel, and that provides basically a ruler that tells us what sizes of DNA we're looking at. How is this visualized? Why are these DNA pieces in this color over here, lit up? What do we add to the gel?
Ethidium bromide.
Yes, ethidium bromide. Thank you. Yeah, we've added a dye, ethidium bromide, that concentrates in the DNA and is fluorescent. Okay, this works really well. This tells us a lot about DNA length and a little bit about its structure. You can look at, for example, supercoiling using this technique. Okay, yeah, here's the ethidium bromide. Here it is intercalated into the DNA. And this in practice is what it really looks like. It has a bright purple color. In addition to separating on gels, we very commonly separate out DNA using capillary electrophoresis. And this is a much more effective way of separating out DNA that differs by a single base in size. So you can take a large number of sequences of DNA and then separate them out such that you have 100 MERS, 99 MERS, 98 MERS, 97 MERS, 96, etc., all neatly separated out using this technique called capillary electrophoresis. It's the same electrophoresis technique with a different mobility layer, different mobility phase, but it's basically using electrophoresis again, but in a capillary. Okay, here's why this is important. You can use this gel-based separation as a way of sequencing DNA. If you had some way of breaking it up into little pieces where each piece differs by one base pair. In practice, what we do is, and I will only tell you about this one over here. Okay, this is the old-fashioned way to do this using these, actually we use acrylamide gels. This is back when I was a postdoc. We no longer do this. No one in my lab does it this way. We all do it this way. Here's the way this works. Okay, what we do is we add a very low concentration of dideoxy terminators. This is exactly what was invented by Gertrude Elion on the Gertrude Elion slide that I showed you earlier. This is missing the 3' hydroxyl. It has a hydrogen in place over here. And these 3' terminators, missing this hydroxyl, shut down DNA polymerase synthesis. When they do, they also carry along a fluorophore that has a specific wavelength associated with it. Okay, so you add a 1% concentration of the dideoxy C inhibitor that has, say, a green fluorophore. You add 1% that has G and has a red fluorophore. You add 1% that has T and has a purple fluorophore, and you see where this is going. So you have 4 dideoxy terminators, each one with its own fluorophore, and you set these fluorophores up so there's no overlap of the wavelengths, or a little overlap as you can get. In the end, you separate out the sequences using capillary electrophoresis, and what you can do is actually read out, based upon the red, green, or blue status of each of those dyes, that you have a sequence that says GAT, CTT, GTT, etc. And in practice, we actually simply take the data, feed it into a program, and the computer gives us a sequence at the end. Okay, so this stuff is massively automated. Okay, in fact, we're at the point where our lab simply sends out the sequences, and there's another lab that used to be on campus, but now it's actually, where is it? It's here? San Diego. Okay, yeah, so it's in San Diego that does all the sequencing for us, and it costs only a few dollars per sequence. Okay, and that price has dropped enormously. Okay, any questions about anything we've seen today? Yeah, Anthony?
Oh, I don't know if you've said, but I'm a little curious about donor and acceptor.
Oh, yeah. Let's put that off. Okay, yeah, fluorescence technology is interesting. All right. Last thing, you can use this DNA technology for all kinds of things. It's used very extensively. You can even use it to program changes in organisms like Drosophila, and you can use this to test hypotheses. It's a little hard to tell, but do you see the extra eyeballs that are growing out of this organism? Eyeball at the stalk, eyeball over here. Yeah, so you can actually program, you can insert sequences of DNA into these organisms and coax them to get turned on at specific times and use this to test whether or not we understand what specific genes do. And this is actually a fascinating story that actually deals with a heat shock protein. Okay, there's a couple of ways of modifying DNA in organisms. One way is to do it randomly, using compounds like this one, whose mechanism we'll talk more about on Tuesday. So let's stop here. When we come back next time, we'll be talking more about this.